Hi, my name is Arshia Kaur, and I'm a student researcher in the lab of Dr. Margaret Riley at the University of Massachusetts Amherst. Today, I'll be discussing with you our research on screening for potential bacteriocin-producing isolates against the pathogen Erwinia amylovora, which is responsible for fire blight. Before we get into the research, I wanted to give you a little bit of background on fire blight and why it's so important. Fire blight is a plant pathogen that affects primarily apple and pear fruit trees. It gets its name from the scorched look that affected tissue tends to have. The bacterium begins in the cankers of fruit tree bark, and as it proliferates, it is able to spread to the other plant tissues, such as the leaves, blossoms, and fruit of the plant. If it's left untreated, the bacteria can survive through the winter and go on to affect the crop year after year. Occasionally, this results in the necessary need to quarantine entire orchards, and a single outbreak of fire blight can affect thousands of acres of fruit trees. Economic costs, which include both losses due to crop loss and prevention, can be estimated to be around $100 million a year, which is really high. There are many treatment options available to combat fire blight, including copper sprays and biological controls. However, the current gold standard, due to the highest efficacy, is antibiotics. Antibiotics have actually been in use to combat fire blight since the early 1940s, and the two most commonly used antibiotics today are streptomycin and oxytetracycline. However, in the previous years, due to overuse of antibiotics, we are seeing a rise in antibiotic-resistant bacteria, and this has affected agriculture and fire blight as well. Due to this increased antibiotic resistance seen in Erwinia amylovora, we see that antibiotics have lower efficacy rates. I have included a couple of studies to highlight this fact. One paper found that a single application of oxytetracycline only provided 60% disease control against Erwinia amylovora. Another study found that 45% of the isolates tested from apple orchards in Utah were resistant to streptomycin entirely. So as we see here, antibiotics are not effective anymore at combating Erwinia amylovora. Antibiotics also tend to be broad spectrum killers and tend to affect not only the targeted bacteria, but also other bacteria in the system. And due to this, antibiotics in use affect the plant growth promoting rhizosphere bacteria present in the soil. PGPRs are microbial symbionts present in the apple's rhizosphere that are essential for healthy plant growth. They are responsible for some functions such as nitrogen fixation and nutrient acquisition, among others. And so it's essential to keep the rhizosphere healthy. We propose a solution in bacteriocins. Bacteriocins are naturally secreted antimicrobial peptides that are extremely specific and effective killers. The purpose of our research was to identify any potential bacteriocin-producing isolates which were effective at inhibiting the lawns of Erwinia amylovora, while also able to preserve the growth of the apple rhizosphere. This was done by screening 220 bacterial strains against the 20 strains of pathogenic Erwinia amylovora and 82 strains of identified rhizospheric bacteria. The rhizospheric bacteria were identified to be from the genera Bacillus, Pseudomonas, Stenotrophomonas, and Enterobacter. The tested bacteria were lawned on a plate, and potential bacteriocin producers were spotted over top. And inhibition was identified by a clear zone of inhibition, which can be identified as the red circle on the photo to the right. Here are the results of our study. In this first figure, I have graphed the percentage of Erwinia amylovora strains which were inhibited by the species we tested. Along the x-axis, you can see which species were active against Erwinia amylovora. And across the y-axis, you can see the percent of the 20 tested Erwinia strains that were inhibited by a given species. The most effective producers against Erwinia amylovora were found to be Acinetobacter baumannii and Streptococcus eani, which were able to inhibit 90% of the tested Erwinia strains. The least effective strains were found to be from Erwinia amylovora itself, which was only able to inhibit 5% of the strains. As you can see here, The bacteria isolates we tested, the potential producers, were actually far more effective than the figures I showed you for antibiotics from previous slides. So what we have is a potential to have a really effective treatment against Erwinia amylovora. So in this figure, I graphed the percentage of rhizospheric bacteria which were preserved by the active strains in an effort to better understand the effect that our strains, which were active against Erwinia, had on the rest of the apple rhizosphere. And as I did in the last figure, all of the active species are across the bottom on the x-axis, and the y-axis is the percentage of the 82 rhizospheric bacteria strains whose growth was not at all affected by the tested bacterial isolates. So as you can see, for the most part, our potential bacteriocin producers did not affect the growth of the PGPRs very much. In fact, 8 out of the 20 active species did not inhibit even a single isolate of the 82 rhizospheric bacteria at all. I mentioned before that Streptococcus eani was one of the best producers against Erwinia, and it was able to inhibit 90% of the Erwinia strains. It was also able to preserve about 94% of the 82 rhizospheric bacteria strains, which is pretty impressive. As I mentioned before, antibiotics are broad spectrum killers, and so they tend to affect the rest of the rhizosphere when applied. But it seems here that the tested bacterial isolates, the potential bacteriocin producers, are not only effective against Erwinia, they have the ability to keep the rest of the rhizosphere healthy, which in turn promotes healthy plant growth. So what can we conclude from these results? We can conclude that these potential bacteriocin producing isolates that we were able to test in this study are able to inhibit lawns and growth of Erwinia amylovora. Of these tested isolates, Acinetobacter baumannii and Streptococcus eani were the best producers, and they were found to inhibit 90% of the tested strains. They are also able to preserve the identified rhizospheric bacteria, which is important because these PGPRs are essential for healthy plant growth. What this all comes together to show is that we could potentially have a viable alternative for preventing fire blights to antibiotics. Our next step would be determining the active agents in these isolates to determine if they are actually bacteriocin producers, and then to conduct assays in apple blossoms and fruit to see how these potential bacteriocin producers would react in the system as a whole. Thank you so much.